First up today is Jie Ma. She is a postdoctoral fellow in Dr. Ken Kane's Fish Health Lab at the University of Idaho. She's been working in the field of fish health and disease since 2008. Her current research focuses on development of fish vaccines, fish immunology, and fish virology. So you can take it away, Jie.
Thank you. Thank you, Dr. Phelps. Hello, everyone. Like Dr. Phelps said, I'm Jie Ma. You can call me Jessie. I've been working on fish health, fish disease research since 2008. I joined Dr. Kane's Fish Health Lab in University of Idaho as a postdoc in January 2017. I'd like to start my presentation here by thanking the organizer for putting the special seminar together, giving us the chance to talk about our recently research, and also thanking our co-author for putting this project together. Dr. Erin Salinas and her lab member, I think they are also here from University of New Mexico. They've been doing a lot of previous research about nasal vaccine and fish immune, especially mucosal immune. So this topic here, long-term efficacy of nasal vaccination against ERM and HNV. This research was based on her lab's previous research. So, okay, let's start today's topic. So let's see the nasal, let me introduce about the nasal vaccine. Nasal vaccine is an effective method to control infectious disease. Actually, nasal vaccine already used commonly in humans and also farmed animals like cattle, piglets, and also chicken, as well as cats and dogs. Because the needle-free nasal vaccine offers offers a novel alternative to the injection delivery strategy, and it has a number of advantage over other type of vaccine. Like it has a strong local immune protection, and it can stimulate systematic immune response like the injection vaccine, and it just needs a low amount of antigen for stimulating the immune response. So based on those advantages, we want to develop the nasal vaccine in aquaculture. I believe all of you in this fish health section know the importance of the fish vaccine in the aquaculture. But we know most of the commercial fish vaccine are injection vaccine. And recently, based on the previous research, we found the nasal vaccine has been shown to be potentially useful in the aquaculture. So the previous research already shown high efficacy of bacterial and viral vaccine delivered intranasal in rainbow trout at 7 days and 28 days after immunization. So in the previous research and this current research, the bacterial vaccine we use is the Yersinia rickeri formalin-killed bacterium vaccine. And the viral vaccine we used is the HNV live attenuate vaccine. So I use the HNV vaccine as example to show the previous research about the short time, 7 day and 28 day post-immunization trials. Like this one showed, like this slide showed in the group A is the unvaccinated group. The fish in this group was given the saline as the unvaccinated control group. After HNV virulence strain challenge, and then those fish has the survival preparation about 35%. And in the second group, the fish was given the HIV attenuate vaccine by intranasal in one air, and then after HIV challenge, the survival preparation was 98% at a 7-day post immunization and 94% after 28 days post immunization. And in the group C, the fish was given HIV attenuate vaccine by the intramuscular injection. And then after the HIV challenge, the survival was 92% or 94%. And in the group D, researchers mixed the HIV attenuated vaccine with the ERM formalin-cured vaccine together, and then gave the mixture vaccine to the fish by intranasal. And then after HIV challenge, we can see here the 7-day post-immunization, the fish survival was 100%, and 28 days post-immunization, they are 92%. And the last group, group E, the fish was given the mixture vaccine by IP injection. And then the fish survival showed 80% or 98% after HIV challenge. So based on those previous research, we can see the high efficacy in the HIV vaccine and for the HIV vaccine and also ERM vaccine by intranasal delivery. So we know, so after got those research data, we want to know what will happen in the long-term response. So we did the long-term efficacy research and want to see if the HIV and the ERM vaccine can give the fish long-term protection after 6 and 12 months after vaccination. So here's this research. We set up 6 groups, T1 to T6, T1 and T4 as the on-vaccine control. The fish was given the intranasal, the fish was given PBS by IM, IP or IN. And in the HIV vaccine group, the fish was given HIV by IM or the intranasal. In the ERM vaccine group, the fish was given ERM vaccine by IP or IN. And then we set up six different treatments, and each treatment have 3 tanks. Each tank contains 130 fish, and the time we vaccinate them, they are 5 gram average body weight. So we collected the fish blood at day 0, 6 months, and 12 months to check the antibody titers by ELISA. We also collected the fish tissue, spleen, and the nettle tissue at 12 months to do the RNA isolation and also the gene expression. Let's see our, and after vaccination, 6 months after vaccination, we challenged the fish by HIV or the recovery. HIV we use is the is the 100 IP injection per fish, and the dose is 1,000 PFU per fish. The recovery we used here is IP injection and 2 times 10 to 7 CFU per fish. In the mock control group, we'll give them PBS injection or the MEM. So we set up 20 fish per tank and 3 tanks per treatment. All the mortality was recorded and collected and examined them. We re-isolated the pathogen and confirmed them by cell culture or bacterial culture and PCR. Let's see our results. Because we raised those fish for over 1 year and tested them 6 months and 12 months. After we finished this research, we based on the data we collect, we found those fish given given iron vaccine or the injection vaccine, they have the good percent survival, 95 to 98%. So we want to say the HIV vaccine and also ERM vaccine are safe to the long-term raising rainbow trout with the iron or injection route. And here is the CPM and RPS after HIV challenge. So after 6 months immunization and HIV challenge, we can see from the unvaccinated control group, the CPM is 96, 97%, pretty high. But the iron group got the lowest CPM is 58%, and the IM got the CPM is 72%. Both of them were significantly lower than the control group. But the difference is from the specific HIV specific antibody titer testing. Because we didn't see the significantly different from those 3 different treatments. But we do see the titer, the HIV antibody titer increased after 6 months and 12 months immunization. So here is the CPM after a recovery challenge. We can see from here, the unvaccinated control group got the highest CPM, 78%. And in the same time, the IP injection group got the lowest CPM and the highest RPS, 70.21%. So that means the recovery vaccine can protect the fish by IP injection very well. And the iron also showed the RPS and with the CPM 63%. Similar like the challenge trial and the specific antibody titer also showed the fish from the ERM IP injection group showed the highest recovery specific antibody IgM titer. That titer is that titer was different with the other 2 treatments. And even at 12 months, there's no difference with those 3 treatments, but we still can see in the iron vaccinated group and IP vaccinated group has the high titer than the PBS control group. So at the last, we also checked the IgM and IgT gene expression. From our data here, we know that our results show after 12 months post immunization, the IgM expression level in the IN HNV group, they still show the higher than the other 2 groups, and in the IN-ERM treatments, the IgM in the fish nose also showed higher than the other 2 groups, the control and the IP injection. But the difference is the IgT gene expression showed negative, showed the decrease. Some of them showed decrease compared to the PBS control group. But most of them no significantly different. So here is the summary of our research. Based on this research, we know HNV live attenuate vaccine and ERM bacterial vaccine are safe to the long-term raising rainbow trouts with intranasal and also injection route. There was similar protection by both the IN and IP vaccination in the HMV challenge. And the long-term ERM vaccine did not show the same level of protection as IP routes. Although low-level specific IgM titer were detected in the sera from the IN vaccinated fish, it is likely that additional mucosal or other immune factors like the IgT are stimulated and play a greater role in such protection. So those points we are still not sure. So we have our future research. In our future research, we want to try if the booster and the adjuvant will affect the nasal vaccine, and we also want to test the antibody changing between 28 days to 6 months. Like we want to check the 2 months, 3 months, and also 5 months to see how the antibody titer dropping. And then we also want to detect other antibodies like the IgT. It might play an important role in the protection. And other vaccine by intranasal like DNA vaccine or other bacterial vaccine. So beyond the vaccine, our plan also want to do other research about the recombined protein like the cytokinase, and then to see how they work with the vaccine. At the end, I want to thank our support and funding again. Thanks to Dr. Serena's lab and also Ken's Fish Health Lab and our project and the University of New Mexico, University of Idaho.
All right, thank you very much.
All right.
Great presentation.
Thank you. Questions?
Yeah, if you have any questions for her, we have plenty of time. You can go ahead and unmute yourself or put them in the chat box and I can read them.
Hey, Jesse, this is Jan. How you doing? Thanks for the
Good.
talk. I was just curious how you arrived on using like a nasal vaccine for mucosal immunity when fish have mucosal surfaces on the skin and the gills and a lot of surface area. So like a lot of immersion vaccines are used for that reason. So how does a nasal vaccine work differently than an immersion vaccine that would target like gill and skin uptake?
Good question. We tested the nasal vaccine because the nasal vaccine only need the small amount of antigen and can stimulate the systematic response and also the mucosal response. So like you said, the immersion can stimulate the mucosal response too, and also for the skin, gill, and other mucosal tissues. So because our research is based on the previous research from Dr. Selena's lab, and then they did the 7-day and 28 days, and then they only checked the spleen and the nose tissue, but didn't involved in other mucosal tissue. So in order to consist their research, just continue, just jump from the short time to the long term. So everything we did just followed their research and didn't involve the other parts.
Okay, thanks. So you would need less amount for probably, I imagine you can use this for bigger fish as well, because those immersion vaccines, oftentimes you target young fry when their immune system's first developed, and you can concentrate them in a small amount of water, so you wouldn't be using as much, but I imagine that this would be more applicable to larger fish. Yeah,
we use 25 microliter per nair, and then 2 nairs, so use 50 microliters per fish. I believe if immersion, that will need a lot of antigens.
Sure. All right, great. Thank you.
Thank you.
Jesse, this is Stephen Reichley. Great presentation as always. Just to piggyback onto Jan's question, I was just curious, you talk about how one of the reasons of going intranasal route is for applicability to industry and vaccinating large numbers of animals. You talk about how the IN route is better than immersion in the sense that it uses less vaccine. Have you worked out, or is someone working out how you physically deliver the vaccine into the nares of large numbers of fish, and particularly small fish?
Actually, compared to the injection, it's more easy. It's more easier to do the intranasal because you don't need to inject the fish. You just need to pipetting the vaccine and then drop the vaccine into the fish there. I think it's a little bit faster than injection, but similar labor work.
Okay, so it still requires handling of individual fish.
Yeah, it still need to handle individual fish.
Okay, thanks.
Thank you.
I got a question. It's Bart from Michigan State University. I would like to know, so when you do the intranasal vaccination, are you just doing one dose and then releasing the fish directly into the water?
Yeah, we didn't do.
Repeated?
No, we didn't repeat, and in this research, we didn't booster them. So that's what we want to do next time. Yeah, because the short time didn't do booster, so we want to consist this research, and we didn't do booster this time. We just gave them one shot.
Not about the booster. It's also because at the end, the antigen is in contact with the mucosal surface for just a few seconds. Then it's widely diluted when you put back the fish in the water. So maybe, yes, you need less antigen, but maybe you need to repeat in order to expose the mucus for more time.
Yeah, in this research, we didn't repeat. We just take those fish when they are 5 grams and give them the vaccine by intranasal, and then put them into the water. The vaccine pretty concentrate.
Yeah, and also I didn't catch one thing. So you say you did mixed intramuscular injection for ERM and IHMV vaccination, but did you also try the mixed intranasal vaccination? So
Yeah. Yeah, actually, those research, actually, that research you mentioned, that research is the previous research. That one didn't happen in our lab, but it's
okay, that was
let me see. So you talked about this one.
Oh, okay, yeah, that
Yeah.
was okay, yeah.
And then the fish was given the mixture vaccine by intranasal and also injection.
Okay.
Yeah.
Thank you very much.
Thank you.
Hi, Jesse. This is Gail Curath. Very nice talk. Thank you. Enjoyed it very much. I always find it really interesting when you directly compare two vaccines and you don't get the same results. So you did get some protection in each, but the IHN vaccine was more protective than the ERM. So my question is, remember, I'm not an immunologist, right? Do you think this means that viral vaccines perhaps work better than bacterial vaccines? Or is this more likely a difference? Maybe if you tried several viruses, you would also have variation. What does this tell you about the probability of success for other viral and bacterial pathogens?
I think there are many factors in fact the final results. First of all, I cannot get a conclusion from this research which one is better, virus vaccine or the bacterial vaccine, which vaccine is better, because there are two different kinds of vaccine. I know you are the expert for the fish vaccine, especially the HMV DNA vaccine, and the HMV DNA vaccine works very well, can go to like 100% protection. But here we use is the HMV attenuate vaccine. So it's totally different. And then then this vaccine show the protection up to 100 by nasal, but most of the time they between 90% in the short time. And you also see here in the long term it showed protection, but not pretty high, not as high as the DNA vaccine. And for the bacterial vaccine, we used here is the ERM formalin killed vaccine. So I think this vaccine is good by the injection. And also this one can stimulate the short time protection by intranasal. So in the short time, it show good protection, but in the long term, it show a little bit, but not as much as the short time.
Yeah, very interesting. I think I don't consider myself an expert on much except IHN. I mean, I don't know the broad. So I do think that looking at different viral vaccines against different viruses, whether they're attenuated or DNA or any form, would be very interesting by the intranasal route. I'm just wondering if the efficacy was because it was IHN or is it because it's viral? I think it'll be very interesting in the future to see how broad this protection is, because it doesn't have to be 100% to be useful in industry.
Yeah. Yeah, so in our future research, we want to test a different kind of vaccine and then collect more research and then to see how the nasal route works compared to the other route.
Yeah, very nice, thanks.
Thank you.
Well, this has been a good discussion, and I hate to cut it off, but we need to switch gears a little bit to the next presentation. Thank you very much, Jesse. We appreciate the excellent talk that you just gave.
Thank you, Dr. Phelps.